Hello everyone, I am Kapish from Chemistry Honours 3rd year from Kirorimal College. Today I'll be presenting on enzymes, the catalysts of life, focusing on their introduction, classification and important characteristics. Enzymes are one of the most important topics in biochemistry because without them, life processes simply cannot occur. Inside every living organism, from humans to plant to bacteria, millions of chemical reactions are happening every second. However, most of these reactions are naturally very slow. If they occurred at their natural place, our body would not be able to survive. This is where enzyme comes in. They speed up reactions so that life can continue smoothly. So basically, enzymes are the silent workers of our body that keep everything running. So, what are enzymes? Enzymes are biological catalysts, mainly proteins, that speed up biochemical reactions. So, I have some key points. They speed up reaction by lowering the activation energy. They do not get used up. After finishing one reaction, they are ready for the next. They work on specific molecules called substrates and convert them into products. So, overall, enzymes make reactions fast, efficient, and controlled. So, why enzymes matter? Enzymes are extremely powerful. They can increase reaction rates by millions or even trillions of times. Because of this, almost every process in our body requires enzymes, like digestion of food, breakdown of glucose to produce energy, growth and repair of tissues, DNA replication, and protein synthesis. Without enzymes, all these reactions would be too slow to support life. So, enzymes are not just important, they are indispensable. So, how do enzymes work? We'll understand this by the lock and key model. The most important thing about an enzyme is that it's picky. It only works on one specific ingredient called the substrate. We use the lock and key model to understand this. The enzyme is the lock. It has a unique groove or pocket called the active site. The substrate is the key. This is the molecule that enzyme needs to change. So the substrate must fit perfectly into the lock, the active site. They bind together to form an enzyme substrate complex. So, the reaction, once bound, the enzyme quickly changes the substrate into new molecules called products and then releases them ready for the next key. So, now we'll talk about its classification. So, enzymes have classified into six main classes based on the type of reaction they catalyze. This classification is done by the Enzyme Commission, also known as the EC numbers. Let me first is the oxidoreductases. It catalyzes oxidation or reduction reactions. Examples are alcohol dehydrogenase. Second type is transferases. It transfer functional groups like phosphate or amino groups. Examples are hexokinase. Third type is hydrolysis. It breaks bonds using water, hydrolysis. Examples are amylase, lipase, protease. Fourth type is lysis. It breaks bonds without water or oxidation. Examples are aldolase. Fifth type is isomerases. It converts molecules into their isomers. Examples are isomerase. Sixth type is ligases. It joins two molecules using ATP. Examples are DNA ligase. So this classification helps biochemists study enzymes in a systematic way. Now we'll talk about the enzyme characteristics like its specificity and efficiency. Why are these helpers so important? Because of these two things. First, we'll talk about specificity. It generally means being picky. Enzymes are highly specific. An enzyme designed to digest fat won't try to digest sugar. This ensures every reaction happens in the right place at the right time. And the next characteristic is efficiency. It generally means being fast. They are super efficient. A single enzyme can process thousands of substrate molecules every second. Now we'll talk about the factors affecting enzyme activity. The enzyme activity depends on the surrounding conditions like temperature, pH, and substrate concentration. Now, first we'll talk about temperature. Every enzyme has an optimal temperature where it works best. If the temperature goes too high, the enzyme can denature, which means it loses its structure and stop functioning. And pH, each enzyme has an optimal pH. Changes in pH can alter the active site and reduce enzyme activity. And when we talk about substrate concentration, if we increase substrate concentration, the reaction rate increases up to a point. Eventually, all enzyme molecules become occupied and the reaction reaches its max speed. Now we'll talk about inhibitors and activators. These have a function of regulating enzyme. So the body needs proper control over enzyme activity. This is done through inhibitors and activators. Inhibitors decrease enzyme activity. They may compete with the substrate for the active site or change the enzyme shape or permanently destroy enzyme activity. This concept is used in many medicines. For example, certain drugs work by inhibiting bacterial enzymes. And on the other hand, activators increase enzyme activity. They bind to other sites on the enzyme, which are called the allosteric sites, and improve the enzyme's efficiency. So enzymes are not only inside our bodies, they are used widely around us. Some applications include like digestive enzymes, amylase, lipase, protease. It helps breaks down carbohydrates, fats, and proteins. And detergents, many modern detergents contain proteases and lipases to remove stains like blood, sweat, and oil. And in food industries, enzymes are used in cheese making, brewing beer, baking bread, and clarifying fruit juices. And in biofuel production, certain enzymes break down plant material into simple sugars that can be converted into ethanol. So enzymes play an important role not just biologically but also industrially. Let me quickly summarize the key points. Enzymes are biological catalysts, mostly they are proteins. They speed up reactions but are not consumed. They are highly specific and extremely efficient. They work best at the optimal temperature and optimum pH. They are regulated by inhibitors and activators, and they have major applications in daily life and industries. Understanding enzymes helps us understand how life functions at the molecular level. So, with this, I conclude my presentation. Enzymes truly are the tiny machines that keep our body and many industries running smoothly. Thank you for attention. If you have any questions, I would be happy to answer.